Hello, and welcome to SNAB Demola. And today's video is something a bit different. I'm going to be trying to go through the SNAB Biology Paper 1 2024. So this is just kind of a walkthrough of the exam paper. I did get a request to do this in a comment. So gonna try, explain all the answers, and hopefully it can be really useful for revision with exams coming so close. So let's get straight into it.
Okay, so for question 1, we're looking here. With the information given us, the first question is telling us to calculate the MPP. So straight off the bat, I know that MPP equals GPP minus R. So now I'm just looking for those values in the question. So they've given me the GPP here. They gave me the respiration here. So I know that literally all I'm going to be doing is 87069 minus 55195. And that should give me the answer D. Okay. Now we look at part 2. The table shows information on the trophic levels within the ecosystem. So you can see the data there. And then on the next page, it's telling us calculate the percentage of the total light energy entering the ecosystem that's fixed as biomass in the primary consumer. So again, I'm just going to look here. In terms of the total energy, we have to go back to the question actually. So this is what it's telling us for the value of the light energy entering the ecosystem. Cool. In terms of the biomass fixed by the primary consumer, it's this value here. So we've used the information we had and we use a table. Now, in terms of actually answering the question, we're going to do percentage. We're just going to do the amount in the biomass divided by the total times 100. So it's 4615 over that 7112800 multiplied by 100. And that will give us our percentage, which is going to be like 0.065% to two significant figures. Always remember that. So it would have been a long answer, but two significant figures. Now, describe the role of mineral ions in the production of plant biomass. So in terms of mineral ions, we're just thinking, what are the examples we know? You just need to be able to rattle these off. Yeah. So mineral ions that we know are important. I know that nitrates are important. I know that magnesium is important. And I know that calcium, as well as another good one, and phosphate is another good one. So you need to remember these and just what they do. Just pick 3, yeah, because it's a 3-mark question. So nitrates, I know, are going to be quite good in terms of DNA synthesis or amino acids or whatever you want to say. Okay. A fact that you should know is magnesium is used for chlorophyll. Another thing you should know is calcium is used for the middle lamella. So I got my handwriting. And then phosphates as well, that's going to be like ATP and stuff like that. So make sure that you do know at least one use of all of these ions because they can ask the question. So again, we're going to look at the question. Anthropogenic activities are increasing levels of carbon dioxide in the atmosphere. So I'm already assuming they're going to ask me about global warming. An increased level of carbon dioxide in the atmosphere leads to an increased rate of photosynthesis in some plants. So they're also going to ask me about photosynthesis. And here, 2A, describe how carbon dioxide is used in photosynthesis. So again, you need to just think, the base knowledge you have, how can you link it to the question? So describe should be fine. They're just literally asking me to just talk about the light independent reaction. So that's what I'm going to start with, light independent reaction. I'm just going to write the acronyms, but you should write the whole thing. So light independent reaction, we know in the light independent reaction, in that first step, you need RUBP to combine with carbon dioxide. So RUBP combines with carbon dioxide, and that helps you to form an unstable 6-carbon compound intermediate. Yeah, so we're saying it forms an unstable 6-carbon compound, and we also know, let's just add more detail, which breaks down, we'll do the arrow, which breaks down into GP molecules. And you know what, just for emphasis, we're going to say 2 lots of GP molecules. Now, I've mentioned a couple of points there. However, I'm just thinking as well, what's another bit of detail I can add on just to sell it? I'll just say that reaction is catalyzed by Rubisco. Typically, if there's an enzyme involved in a reaction that you know, such as Rubisco, just mention it. Okay, so they're giving us more information about the carbon dioxide and the photosynthesis in soybeans. So reading that information, we have an investigation here. So soybeans are in 6 different containers. There's different concentrations of carbon dioxide. Then they weighed it and they did the mean dry mass. So we can see all of that in the table here. Now, it says which of the following describes the relationship between an increase in carbon dioxide and a change in dry mass. So I'm seeing down the table we're having an increase in carbon dioxide, and you can also see that we are having increasing mean dry mass if you just look at the trend. So you're already thinking that's probably positive correlation. Now exponential is a bit different. Exponential means it goes up like this. So we'd have to see, not a tick, but you get what I mean. So we'd have to see a much bigger change if we went exponential, and then you know this isn't negative and there isn't no correlation. Now for part 2, it says the measurements were repeated at each concentration to calculate the mean and standard deviation. Why was this done? So instead of a chi-squared, we wouldn't really do a chi-squared for this, and you also don't need to do repeats for chi-squared, so I can rule that out. In terms of improving accuracy and validity, these are very much method things, which is why you would kind of land on B. So if you do the mean and standard deviation, the reason why that's good for repeatability is you can just see, is there like big differences between the times we did it? Like, do we have a massive standard deviation? If so, probably not that repeatable. So just thinking about stuff like that. Okay, part C. So enzymes catalyze some of the reactions involved in photosynthesis. Explain why an increase in carbon dioxide concentration above 1000 ppm may not increase the rate of photosynthesis. So I'm thinking we're being asked here about factors controlling enzyme in controlled reactions, or factors affecting enzyme controlled reactions. So they want me to talk about carbon dioxide. So what we'll say is carbon dioxide quite clearly is not the limiting factor. So I'm looking what might be, probably the enzyme concentration, because we're not talking about changing the concentration of Rubisco, right? So what we can say is all active sites, key terminology, are occupied. So if all active sites are occupied, that means that our enzyme concentration is the limiting factor. That's the next thing we can say. Enzyme concentration is the limiting factor. Okay. So now that we've discovered that enzyme concentration is a limiting factor, what can we say? That means that if it's a limiting factor, that means it's limiting the rate. So this limits the rate of photosynthesis. That's kind of what I'd be looking for to send the answer. So you're identifying, okay, if it's not carbon dioxide, what is the limiting factor and why? So it will be the enzyme concentration, and it will be because the active sites are occupied. I wouldn't go on mentioning anything like temperature or light just because there's not really any mention of it in the question. So I don't think it's that relevant. But yeah, you're just kind of going on this kind of trend. Okay, question 3. So we are now talking about clinical trials, right? And they are specifically talking about William Mevering. So unfortunately, these questions are very much, if you know it, you know. If you don't, you don't. There's not really any other way about this. So in terms of William Mevering, what you should know is he did his experiment with digitalis soup. So that would be the first thing, digitalis soup. That's what he was testing. And what you should also know is that he was just, well, he used it from Foxglove, right, as well. But he just gave them the digitalis soup. So he didn't use a placebo. He knew he was giving the digitalis soup. There wasn't really anything else that he did. So it wasn't double blind. And he only did it on the people that were ill. So wasn't tested on healthy volunteers, not double blind, not placebo. However, what he did do is if he thought the dose was too high to a point it was toxic, he would reduce it. So he did test the dose, in all fairness. But this is very much just a knowledge. If you know it, you know it. If you don't, you don't. Okay, now for part C. So they're talking about a drug that they're using for cancer treatment. And they're talking about a bit about what cancer works, and they're saying that this drug specifically prevents the spindle fibers from changing length. So when I hear spindle fibers and cell division, they want me to talk about mitosis. They've been sneaky about it, and that links as well to the cancer because they're having a really fast rate of division. They want me to talk about mitosis. So I'm thinking, where does spindle fibers come into the mitosis process? And that's going to be during mostly anaphase, right? So what's actually happening? So we're going to say spindle fibers used in anaphase. Okay, and what do they do in anaphase? They separate the chromatids. Okay, so we're knowing that spindle fibers separate the chromatids. Now, we need to think, okay, that's our point. What's the consequence of that not happening, right? What if the spindles don't shorten and they don't separate the chromatids? Then that means that, one, the chromatids, so anaphase isn't going to happen essentially. Anaphase doesn't happen. And if anaphase doesn't happen, then that means that the chromatids don't go to opposite poles. And if the chromatids don't go to opposite poles, then that must mean that telophase isn't going to happen. So telophase can't happen because they're not on opposite poles. Hence, mitosis doesn't occur. And that will be how you want to answer that question. Okay, so now we're talking cystic fibrosis. It's an inherited condition caused by a mutation in the CFTR gene. Now, the first question, part A, is saying, describe how a mutation can cause a change in the primary structure of a protein. This is one of my favorite questions in A-level biology because it's the same every time. So I'm going to just use a symbol. You have a change in the DNA base sequence, and we'll follow a logical chain. So change in DNA base sequence, which causes a change in RNA base sequence. If there's a change in the RNA base sequence, then that means that there's a change, so mRNA, I should say. They typically won't ask to do RNA, will just be mRNA. Then that means there's a change in the sequence of amino acids. And that should be enough because your primary structure is a sequence of amino acids joined by peptide bonds. So that alone should be enough to get you all three of those marks. Now, the next question, CFTR is produced in the cells in the lung and is involved in the transport of chloride. So describe the location of CFTR in the cell membrane of one of these cells. So again, quite a simple question, I would say. In terms of protein structure, you should remember you've got your phospholipid bilayer, and then your proteins or your channel proteins, which CFTR is, tend to just be embedded like along both sides of the membrane. So they kind of go like that. We'll just do another one here. And then you still have your phospholipid bilayer. So that's what you really want to put into words. So just say that it's embedded in the phospholipid bilayer. And it spans across both sides of the membrane. And that's what you want to say to get your marks there. Another one of my absolute favorite questions, but we're talking about now CFTR being transported from the ribosome to the cell membrane. So essentially, we kind of want to talk about, first of all, what's happening in the ribosome. You need to position yourself. So know that translation has just happened. So if we have had translation, we are a primary structure protein. Now, where does it go? It goes from the ribosome to the rough endoplasmic reticulum. And what's going to happen there is going to get folded into the tertiary structure. So that's the first thing that's happened. Then what happens after the RER? Well, it's going to go from the RER to the Golgi. And at the Golgi, that's why you're going to have modification, really. So modification, and I always like to throw in an example, E.G. Addition of carbohydrates chains, just to kind of make my answer stand out a bit more. And then what I also say, and packaged into vesicle. And once it's in the vesicle, what's going to happen? Well, the vesicle is going to undergo exocytosis. So vesicle fuses with the cell membrane. And then the result of that is exocytosis. Okay, so that's the first one. And then for C, it's saying the most common mutation in CFTR makes a protein has the incorrect shape. So why does the change in the primary structure of a protein affect the 3D shape? So let's think. Primary structure, what's that? A sequence of amino acids joined by peptide bonds. So that's what I'm going to say. Sequence of amino acids And again, linking to the question we just answered, that means that we have changes, yeah? So the sequence of amino acids changes. If the sequence of amino acids changes, then that means that the R groups, very important whenever you talk about 3D structure, R groups are in different positions. So my handwriting again. So if the R groups are in different positions, then that means the bonding between R groups changes. So what we'll say is that different bonding or different position of bonding between R groups. And here I would always throw in an example. So for example, disulfide bridges or ionic bonds are quite good to mention. I typically mention 2 just to be safe. And then finally, if we have that, that means that protein folds into a different shape. If it's an enzyme question, specifically mention that the shape of the active site changes if it's relevant. But those are the things that you want to say for that question. Okay, question 5. So we're talking about photosynthesis again. Now, this question is purely just knowledge. Like, these are things that you should know if you're doing A-level biology. So, first thing we're saying, labeled L, well, you can see here, L is a thylakoid. That's just something that you need to know. So, light-dependent reaction happens in the thylakoid, that's fine. J, you need to know, is your stroma. So, matrix is going to be in a mitochondria. This is a chloroplast. So, we're talking about stroma. And we know in the stroma, the light-independent reaction occurs. That's something you should know. So, we're happy with that. And then finally, what's stored in the structure N? Well, N, again, you should know this, is an amyloplast. And amyloplasts store starch. So they're actually going for D for all of these, but those should be your answers. That's just knowledge marks. Okay, so for B part 1, again, knowledge marks. These are things that you should know coming into the exam. So in terms of products of the light-dependent reaction, you need to know oxygen, you need to know ATP, and you need to know NADPH. Those are things that you must know going into the exam. Now, in terms of part 2, this is now an experimental design. So we're looking at the light-dependent reaction, and we're looking at DCPIP, and specifically its decolorization. This is a practical that you should know. It's one of the core practicals. So in terms of what we do for this question, first of all, let's kind of think about the method that we want to use. So what I know is we need to have test tubes containing DCPIP and chlorophyll, or in chloroplast, I mean. So we need to have those test tubes set up. And what we're going to want to do is we need to kind of let the reaction occur, the light-dependent reaction, and we need to kind of investigate what factors. So we're looking at temperature. Cool. I'm just orientating myself. So in terms of something that we definitely need to do, well, we need to make sure that we think about our variables. So the main variable we need to control is light intensity. They will 100% want you to say light intensity. So because light intensity affects the rate, so light intensity must be the same. And we can do this by like putting a lamp or a dark room or something like that. Not a dark room really, not for this. But yeah, putting a lamp will help you to control the light intensity. Now, we also need to think about other variables. So I think it would be a bit remiss of us to just put one. So I'm thinking, what other things can we do? Well, the pH of the solution is a good one. The volume of DCPIPs. You just need to be thinking about these things when you have a practical question. And then also, for this one, because I've seen mark schemes like this before, I know they're going to want us to put biotic variables as well. So for example, let's say the plant species that you got the chloroplast from. So you're just thinking about stuff like that. And the more you do past paper, the more that you'll see what kind of things to say in a question like this. And then finally, well, it's all good doing that, but we need to think about how we're actually going to test the rate of photosynthesis in this. Well, you just need to see the time taken for DCPIP to decolorize. And apologies, I completely forgot to do one thing. So we set our control variables. We also need to think about the independent variable, which is obviously the temperature. So I've just mentioned your dependent variable there about the time taken for DCPIP to decolorize. However, in terms of your independent variable, the temperature, you need to give a range. In SNAB, they love ranges. So I'll always say a water bath is always a great shout. So test tubes go in a water bath. And then they want you to say a range. So don't do anything ridiculous. Like if you say 100, they're going to mark it wrong. But just think, I think like 0 to... I wouldn't go higher than 60. I wouldn't go higher than 60. But 0 degrees to 60 degrees should be about fine. But yeah, always make sure you're doing different ranges. Okay, so they're talking about climate change. I was hoping it was going to come up at some point based on the earlier question they said. But they also want to talk about germination of plant seeds. So I'm thinking they might talk about sustainability as well, or seed banks. Now, first question you've got here is about the structure of starch. This one is a warm-up. This is something that you would have learned in year 12, and I think you should know going into the exam. So in terms of structure of starch, what we really want to say is it's a polysaccharide, right? Polysaccharide of what? So it's a polymer of alpha glucose. That's the first thing to mention. Yeah. So polymer of alpha glucose. Now, what else do we want to say? Starch specifically is made up of two, right? You should know amylose and amylopectin. Yep. You also want to make more comments. So in terms of amylose, this is unbranched and it contains alpha 1, 4 glycosidic bonds. And then the other guy, amylopectin, is branched and it has alpha 1, 4 and alpha 1, 6 glycosidic bonds. So you're making sure you're mentioning all of that in your question and you should get remarks. Okay, so it says starch in seeds is broken down by enzymes during germination. I'm just going to highlight early doors. They've mentioned enzymes. Now, an investigation into the effect on temperature and germination was carried out. They used seeds in different species of North African woodland plants. And then you've got the results down here. Okay. And we've got mean spring temperature plus 5 degrees. Okay, that's fine. Now, explain the effect of an increase in temperature on germination of the seed. So, first thing I'm going to say, explain. If I have an explain question, that means that they want me to pick out a piece of data and then they want me to use my biological knowledge to tell them what's going on. So that's the first thing I thought about. Now, in terms of increase in temperature on the germination of these seeds, I'm looking at the data. Now, what I can see is in the mean spring temperature plus 5, it's higher for every single one of them, and that's significant. So because it's high for each one, that's what I'm going to start with. Increase in temperature leads to an increase in germination. So we've said that straight off the bat. Now, why? If you increase temperature, again, we're thinking back to enzymes. This is something that you would have been doing throughout your course. If you do chemistry, you'll do it as well. So there's increased kinetic energy. So not enzymes, energy, which causes more frequent collisions. If we have that, then that means that we're forming more enzyme substrate complexes. So increased enzyme substrate complexes, which means that you're going to increase the breakdown of starch. And if we increase the breakdown of starch, well, just think, how does that actually relate to germination? That's the last thing I kind of want to touch on with this explanation. Well, starch is made up of glucose. If we have more glucose means that we can increase the rate of respiration, which will provide the energy for germination to happen. So those are what you want to say in that. Okay, so looking at these two questions, I'm not going to lie, this is purely just practice will mean that you'll get this mark. Yeah. So global warming, climate change, said it was coming. Now, in terms of distribution of plants, every single time that you're talking about climate change, you want to talk about temperature and you want to talk about rainfall. Those are the two things you need to mention. So in terms of temperature, we've seen it. I want you to do this. Increasing temperature will cause earlier germination. That's what we've seen in the graph earlier. Okay. And now, in terms of rainfall pattern, I would just say a general statement in terms of rainfall pattern can cause change in the plant's distribution. So change in rainfall can cause change in distribution. And that sounds like a very vague point, but literally, whenever you have climate change, they want you to talk about temp and rainfall. Definitely. Cool. Now, in terms of seed storage, seed banks, they said cold and dry conditions. And again, if you've done a bunch of questions on seed banks, you'll know what they want you to talk about. So, first of all, we're thinking, okay, let's think about relation to germination of seeds. We want to prevent germination. That's the whole idea of the seed bank, right? So, cold, dry, especially cold, you're thinking lower kinetic energy. We're going to cold leads to a reduction in enzyme activity. And if we have a reduction in enzyme activity, then we prevent germination. And then we've addressed one of those things, we've addressed cold, so let's now address dry. What you should know is dry leads to a lack of bacterial growth. And that should get you all 3 marks. I think the points will be enzyme activity and the prevention of germination, and then the last one will be about bacterial growth. Okay, so they say deer are herbivores living in the wild and in the reserve, and you can find several species of deer in the UK. Cool. Adult deer have no natural predators, and deer populations are increasing. So, in terms of population, again, this is definition. So you're just looking at them. You're saying that a population is a group of individuals of different species living in the same habitat. Nope. Of one species living in a different community. Nope. Of one species living in different ecosystems. No, but they are a group of species, a group of one species that lives in the same habitat. So I'm happy to do D. Now, in the wild, several species of deer can live in the same area. Why? So you're just thinking again, you need to use your knowledge. So in terms of animals coexisting with each other, etc., each species has its own niche. So each species has a specific niche. And given that, that means that there's no competition. So if they each have a niche, that means that there's not competition. So no competition. And then honestly, again, with this spec, they love an example. So no competition for what exactly? So I'll just say for food. Even if you think that's a bit of a stretch, that's what you just need to focus on saying. Now, let's look at question B. In one species of deer, mutations led to the occurrence of a white deer. In some reserves, white deers increase in number. So they've given us the data there, 550 and 250. And they're saying that the color of the coats is controlled by a single gene. Very importantly, they've told us that brown coats is dominant when my highlighter works. So brown coats is dominant and white coats is recessive. Good to know. And we need to use Hardy-Weinberg equation. So again, you need to know this equation. But brown was, oh my goodness, brown was 250. No, sorry, brown was 550. And white was 250. So I'm just thinking again, in terms of what we know, I can do Q squared because Q squared is the number of homozygous recessive, or the frequency of the recessive allele. So I'm happy to do 250 divided by 800 because that's the frequency. So 250 divided by 800 gives us 0.3125. So 0.3125 is equal to Q squared. So we can just square root that, root of the answer. I'm going to round. Q is 0.56. Now we know that 1 equals P plus Q. So rearranging that, we can find P. 1 minus Q equals P. So P, we're going to have an estimate of 0.44. So we found P, we found Q. Now I'm going to look at what they wanted. They wanted the heterozygotes, which is given by 2PQ. So I'm happy to just do 2 times 0.44 times 0.56. Now, in my calculator, what I'm probably going to do is do answer multiplied by 1 minus answer multiplied by 2. And that's given me 0.493. So if that's the case, then to find the number of heterozygotes, I just need to do that times by the total amount, which was 800. So we'll do 0.493 multiplied by 800, and we'll see what we get. 394. So that's what I'm going to put on the answer line, 394 individuals. Now, we're looking at part 2. So it said it might be beneficial for a population to contain a high percentage of heterozygotes. Interesting. Let's just think about the statement, is it true or false? They love these questions in SNAB. So heterozygotes have an allele, which allows for adaptations for conditions to change. I would agree. Just think about it, because they have both, it means that they are more adaptable compared to just having one. Heterozygotes reduce genetic diversity? No, because they have both alleles. So that means that you're having more alleles in the gene pool. Okay. Beneficial allele could be recessive, and heterozygotes being together can produce homozygous recessive. That's very true. So I'm happy with 1 and 3 being the true statements. Okay, so we have a practical question. They're looking at a study, grazing by deers reduces plant biodiversity. Okay. And an area with a large population of deers compared to an area where there are no deer. And we need to compare the biodiversity. So we're thinking topic 5, core practical. Now, first of all, you're going to want to set out a grid. So you set out a grid of the same size. Okay, fine. Let's say like 100 meters squared. I just think it's always good to just put an example that's appropriate. So now that you set out a grid, what you want to say is you need to kind of throw your quadrate into those places. You need to create randomness. Again, if you've studied the practical, you'll know. So you need to like maybe do a random number generator to pick coordinates. Yep, because throwing it isn't actually random. So random number generator to pick coordinates. And then that is where you're going to put the quadrant down. So put quadrant, and again, you're thinking key terminology, equipment. So you put the quadrant in those coordinates, or at those coordinates. My apologies. Now, you need to do this definitely at least 10 times in each area. At least 10 times. Again, studying the practical, you'll see that you need to do it a lot of times. So repeat 10 times in both areas with the different coordinates. Okay. I think also sometimes they like you to just mention the size of the quadrant. So I might go like 1 meter times 1 meter, given that we've got 100 meters squared to work with, right? Now, we've kind of done the procedure, but we need to think about how we're actually going to measure the plant biodiversity. So procedure is done. Plant biodiversity is the next thing we need to cover. Now, you need to count the number of species, or the number of plant species in each quadrant. And then on top of that, count the number of each species. So you count the number of species, say there's 5 different species, then you count the number of plants in each species. That's a better way of putting it. So I'll hand out again. Okay. And I think some of you would have realized why we're doing it. It's because we want to calculate an index of diversity. So calculate an index of diversity. And that, my friends, should be good for that practical. Yeah, I think sometimes they might also like allow you to do a t-test. I think some people will just focus on doing the species richness, which is okay, but just because they said plant biodiversity, I think you should focus on doing an index of diversity. Okay, so they're saying DNA from skeletons of people living in the UK over the last 4,000 years has been studied. Okay, and one of the gene studies is lactase. So I'm thinking this might end up being about evolution, but we'll see where it goes. Now, which of the following statement about the action of lactase is true? Just think about it. Lactase, they've told you, is an enzyme. So what do enzymes do? Enzymes lower the activation energy without being used up. So the correct answer is D. I feel like all the more are going to do. Anyway. Discard the role of ribosomes in the synthesis of lactase. Now, I'm just thinking, synthesis of lactase. Lactase is a protein. So a hidden question. They're essentially asking you to describe translation. That's what they're asking you to do. So let's go through it. We're going to say that mRNA is made in the nucleus. Okay. What happens? Well, mRNA binds to the ribosome. Okay. Now, what happens with the ribosome? Well, the ribosome is going to read the mRNA in groups of 3 called codons. So that's what you want to say. mRNA reads, ribosome reads mRNA. My apologies. mRNA in groups of 3 bases. Which is called codons. Okay. After we've said all of that, we're thinking we need a tRNA molecule. So tRNA with complementary anticodons. What does it do? It brings an amino acid. And just remember, complementary is a really important word there. Now what happens? The ribosome is just going to move along. So ribosome moves along mRNA until the stop codon. And for the piece de resistance, we're thinking, okay, synthesis of lactase is a protein. Well, the end goal is to make a primary structure protein, which is a sequence of amino acids joined by peptide bonds. So I'm just going to mention peptide bonds form in, I'm just going to add in condensation reaction. Between adjacent amino acids. And then that should guarantee the 3 marks. Okay, so they said in most mammals, the production of lactase enzyme decreases when the offspring stop feeding on milk. In Europe, a single mutation results in the continued production of lactase by children and adults. Okay, and they've given us a table of the mutations frequency over 4,000 years. So they're talking about the percentage increase in Central Europe, and they want us to do the same for UK. So I'm just looking, first of all, let's just look at this side of the table. And we're just thinking, okay, well, present day is 0.74, before it was 0.096. So just get those bad circles out. But you're just thinking it's new minus original divided by original times 100. I'm just thinking, okay, pop that into my calculator and I should get the answer. So the answer that you would get is about 671%. Okay, so this said that during the last 4,000 years, changes in farming led to a greater increase in the consumption of milk and dairy products in the UK compared to Central Europe. So that many children in Europe have a mutation to make the continued production of lactase. So comment on the effect of consuming milk and dairy products on the change in the frequency of the mutation. When I see a change in frequency of a mutation, they want me to talk about natural selection. That's almost a guaranteed. So whenever we have a natural selection, we're first of all thinking that there was a random mutation. What is it for in this case? For production or for continued production of lactase. Okay, that's number one. So we've got a random mutation. We need to think, what was the selection pressure? Well, the selection pressure would have been the fact that they are still having milk and dairy. So we're going to say that milk and dairy, or the diet with milk and dairy, is a selection pressure. So because of that selection pressure, it means that the lactase allele is advantageous, because these guys are obviously going to be able to get better nutrition because of their diet. So that's what we want to say. And now, if they have an advantageous allele, let's link it, more likely to survive and reproduce. If they survive and reproduce, what do they do? Reproduce. If they do that, that means they pass on advantageous allele to offspring. And if they've done all of that, then that means that the frequency of the advantageous allele increases. So over time, increased, or just do increase allele frequency. So that would be how. It's a comment on question. So we've used information in terms of, we're thinking, okay, well, in Central Europe, they're having more milk and dairy. Oh, sorry. In the UK, they're having more milk and dairy. Yeah. So that will be why the percentage increase is greater. I hope that makes sense. Okay, so they're talking about hepatitis C, which is a virus that infects the liver, and some children with hepatitis C get very bad liver issues. So, fever is one of the symptoms. Now, what is the role of a fever in the immune response? So, we're first of all thinking, okay, is this non-specific or specific? Non-specific. Yeah. So, non-specific. Okay. Immune response. And what's actually happening in a fever? Well, you should know, because I'm sure everyone's had a fever here before, that you increase the body temperature. I would like to say core body temperature more specifically, but you're increasing body temperature. Okay. Just think about it. Why is increasing the body temperature useful? We've talked about it all throughout this question paper. It's probably going to be about causes enzymes in the pathogens to denature. That's what I would say. Causes enzymes in the pathogen to denature. That would be the angle I would go for. But obviously, there's different angles you can go for. I think if you just say that the higher temperature is going to destroy the pathogen, that would be fine. But make sure, I didn't write it here, but make sure that you're saying higher temperature causes enzymes. Yeah, be very specific with your wording. Now, in terms of describing how activation of T helper cells leads to production of antibodies, again, this very much is knowledge, something that you just need to memorize. So, you know that your T helper cell releases cytokines. And those cytokines are going to stimulate, or we'll just be specific with the language, cytokines stimulate B cells. Okay? So when your B cells are stimulated, B cells become, or they produce, B effector cells. And we know that our B effector cells secrete, or they differentiate, we should say, differentiate is better, into plasma cells. And then we can use that word secrete. So plasma cells secrete antibodies. And again, this just comes from knowing the topic. So that question, if you've learned the topic, shouldn't be too bad. Because the hepatitis C enters the liver cells and then destroys them. The virus causing hepatitis C has a similar structure to HIV, which is an RNA virus. Okay. Now, literally, that means that they want you to talk about HIV. Like, of course, you're saying it in the context of hepatitis C, but it's going to be exactly the same as how HIV enters cells, which means that you need to use your knowledge about HIV that you would have learned in the course. So, remember, in HIV, it's GP120 binding to CD4. We're literally going to say the same thing. So, glycoprotein binds to a, remember, complementary, always complementary receptor on liver cells. Okay, so now we're saying the hepatitis or the glycoprotein on hepatitis virus has binded to a liver cell. Okay, now again, what happens in HIV? The viral envelope fuses with the liver cell membrane, and then that means that the viral RNA can enter cells. So again, very much the same as HIV, but now you're just saying it in a different context. Okay, so we're not going to actually write a 6-mark response because that will take too long, but we are going to kind of talk about what this question wants you to do. So first of all, when I say 6-mark, I'm thinking discuss is a command word. So what does that actually mean? Well, it means we need to identify the issue. How do we identify the issue? We want to explore all aspects. And finally, we want to kind of give our opinion. That's what they kind of want you to do in a discuss question. So let's see, first of all, what are they kind of going to be doing here? So we're talking about hepatitis B now, and we're talking about the different treatments. So essentially, what they're going to want to be doing is saying, okay, does drugs, antibodies, vaccine, arguably, which is the best? Arguably, that's what they'd want you to say. Yeah, they want you to compare all of those in terms of their effectiveness in terms of managing a patient with hepatitis B. So we're thinking, okay, I would like to just kind of separate them out and talk about them each individually. So if we talk about drugs, we're saying that drugs can definitely treat an infection that's happened, right? So for someone who's already had an infection, drugs will be good. That's one thing to say. So drugs treat infection. Okay. Now, what else can we say? Well, we know about different types of immunity, don't we? So we know that antibodies is artificial passive, and we know vaccination is artificial active. Of course, these are longer term treatments. They are longer term treatments, but we know they provide different types of immunity. I'd be getting that down. Another thing that you'd want to be talking about is, okay, let's compare vaccination versus antibodies. Well, vaccination will make the memory cells. The antibodies won't, because your body has to fight off that attenuated pathogen, right? That's another thing to mention. What else can we say? Well, in terms of the drugs, I would maybe do a comparison between the two. So if we inhibit the viral polymerase, what does that mean? Well, it means that the actual virus cannot replicate. Yeah, it can't replicate, it can't spread. Whereas the interferon doesn't do that. We should know about what interferons do based on our own knowledge. So your interferons are likely to inhibit protein synthesis or the formation of new viral particles, but they do not actually prevent the replication and spread. Hope that makes sense. Now, in terms of how we can kind of think about scientific knowledge, because they want a sustained line of thinking, you need to be mentioning things throughout that show the examiner I know about this biology. So we're thinking about things like viral load need to be mentioned at some point. We're thinking about how do vaccines and antibodies actually destroy a virus compared to the drugs. We're thinking about maybe things in context. So like, for example, if a newborn baby is like immunosuppressed, interferons would be really good. That's an example. You're also thinking, okay, if I work in a hospital where a lot of patients getting hepatitis B, the vaccine is going to be good for me, right? Because it's long-term protection. I'm going to be working in that environment for a long time. And then you're also thinking, okay, drugs. If you've been infected, exposed, give them the drugs. That will be better than giving them a vaccine at this moment in time. So you just need to think very, very contextualized about these points, and you need to be trying to link them all together. Another thing that maybe I didn't say as well is your vaccine, you need to get 3 doses. Yeah, drugs, a daily tablet you just pop in your mouth, I assume. So those are just different things. Injections are a bit harder than just taking the vaccine. I would say injections are a bit harder than just taking an oral tablet, stuff like that. So I unfortunately, I'm not going to be able to write down a full answer for you to have a look at. But I hope just talking about those points is kind of giving you a bit more perspective into what kind of things you want to be putting down on the paper when answering a question like this. Okay, so last question. We're saying resistance of bacteria to antibiotics is an increasing problem, and we need to make new antibiotics. So it's probably going to be that practical, one of the core practicals. Now, they said the researchers developed a new antibiotic effective against multiple strains of bacteria. They are all bactericidal. So bactericidal, we know, kills bacteria. Bacteriostatic is a different one, or bacteriostable, and they prevent the reproduction of bacteria. So you can see here, B is going to be your answer here, whereas C would have been if it was bacteriostable. Now, we actually go into the practical. So they've developed antibiotic A and B, and they are tested against 2 species of bacteria resistant to ciprofloxacin. So they're kind of describing the practical method here. We might have to go back to this later, but I can see the results here. So what I'm going to ask is the mean rate of decrease in the number of cells in the first 4 hours for species P and antibiotic P. So I'm just looking here, species P, let's look at this graph. Antibiotic B is the squares. So in the first 4 hours, we need to cut it off, it's here. So again, you're thinking, okay, 10 squares for 5. That means each of the squares is like 0.5. So that's why we're going to get 4 cutting off there. Then we're thinking, okay, let's see what it was at 0. So my iPad's about to die. But we can see it's like here, which is like 6.8-ish. because this is 2 and 5, so each one is 0.4. So we know that at 4 hours it's gone down by the log base 10 of 6, log base 10 of 6.8. Yeah, so we need to do anti-log. So what you're actually going to do is 10 to the power of 6.8 divided by 4, and that should give you the answer. So 10 to the power of 6.8 divided by 4 gives me 1577393. That would be the answer. You can make it a bit easier for yourself and like maybe say it's like 1.57 million or something like that. I think you can get away with like 1.577 million. But I would probably just put the big answer on my thing and they'd be fine with it. Okay, so we're commenting on the effectiveness of antibiotics A and B against both species of bacteria. So I'm just gonna kind of rub off what I put here. So when we're commenting on, we need to kind of get all of our data together and then make a judgment. So I'm just thinking, okay, P, let's see. Well, for antibiotic P, I can see that in terms of eradicating the bacteria, antibiotic B has done a better job than A. So that's the first thing I'm going to say. Antibiotic B is more effective against bacteria P. I'm just looking at Q. We actually maybe have a different story just because for B, it's gone to 0 quicker, but then some of them kind of got back up, so there's some resistance there. Whereas with A, although it took longer to get there, it kind of stayed there. So I would first of all be mentioning, okay, so A is better against, sorry about my handwriting in this video has actually been atrocious, against Q, and then B is better against P. Okay. So those are things I can say. Now, what I can say is that we definitely want to mention this, right? We definitely want to mention this. So in terms of what happened with Q, I would definitely mention that the bacteria are not resistant to antibiotic B, definitely not. Yeah. So I would mention about not, oh, they are resistant. So yeah, I would say that they are resistant to B, or bacteria or Q is resistant to B. I would maybe mention specifically that that's happened at low values. So at low values. So sometimes you have like high dose, low dose, or like when there's a high load or low load. So I would mention that. And another thing that I'd want to mention as well, just a comparison between the two graphs themselves, is one of them better? Are they both better? They're both better in Q because they get to 0 faster for Q. So yeah, I would also mention that A and B are both more effective against Q. So yeah, those are things that you need to look at when you're answering a question like this. Okay, finally have the serious question on the practical. So they said the further investigation found the optimum concentration needed to kill bacteria for each of the new antibiotics using cultures on agar plates. So for each antibiotic, we need to do a procedure to find the optimum concentration. Now, first thing that you're going to think, okay, well, I need to think about this practical. It's a core practical. What was really important? Aseptic technique was pivotal. So we need to talk about aseptic technique. So this will be about like autoclaving or making sure your things are clean. That's something that needs to be mentioned. Now, we need to have agar. So we'll talk about equipment first. So we need agar plates with the bacteria. And we want to make sure it's the same bacteria in each one. So that's a control variable that we've mentioned. Now, what we need to talk about as well, what other kind of things do we need? Well, I'm going to just say about autoclaved equipment. I've kind of forgotten which it is, specific equipment, but I don't need to know this anymore. So you should know the exact equipment, but make sure you're saying something like autoclaved. Now, what we want to do is we need to get different concentrations of each antibiotic. So that's something you need to mention. So now we're talking about variables. So different concentrations of each antibiotic. That's important. And then something else that we're probably going to need to say is, okay, we've got our independent variable. What about control variables? Same temp, same time, same length of time. And obviously we mentioned about same bacteria, so let's just put a biotic one in there as well. Same species of bacteria. So those are all good now. We're building our case. Now, we talked about that. We talked about the control variables, independent variables. What's the dependent variable? In this one, it's to measure the zone of inhibition. And that will tell us about it. So you can actually kind of do this in like bullet points where like you've told about your variables, you told about an equipment list. But after you've done that, we need to probably now talk about, okay, let's talk about practical. So in terms of the technique that we're using here, well, we have agar plates with the same species of bacteria, autoclave the equipment, and then what you need to do is we need to incubate it. So we have an incubator at the same temperature. You can mention the temperature. So like, I think a good one here might be like maybe 25-ish degrees or something like that. Yeah. And then for length of time, just mention about like maybe 24 hours, I think is reasonable. And then that's fine. Okay. What else do we kind of need to measure? I would say after that, so of course I haven't written down about the actual procedure, but you should know to write down about that. And then finally, I'm going to talk about in the end, okay, repeats. So we want to repeat the experiment a lot for each antibiotic. For each concentration, actually. We are going to repeat for each antibiotic, but talk about for each concentration. So we want to repeat, and then we probably also want to calculate a mean zone of inhibition. Those would be good things to say. I don't even mind as well, just saying like standard deviation, something like that might be good as well. And then furthermore, if you really now want to kind of push it to the upper echelons of a response, mention things about repeating with like smaller concentration intervals. So if you've kind of found, okay, my optimum dose is between like 0.5 and 0.6, so just put about to that. Say if you find it's between like 0.5 and 0.6, then you start saying, okay, let's do 0.55, let's do 0.56, really kind of narrow it down. And then you can find the minimum effective dose, because you definitely want to use the minimum in terms of bacteria, in terms of antibiotics. But those are the kind of things that you, again, need to be thinking about. I'm not going to go and write an answer because it will just take ages. But those are the things you need to think about when answering this question.
Okay, thank you so much for having a listen to that video. I hope it was helpful in terms of having a student who did get an A-star in the A-Level SNAB Biology to kind of go through the thought process in terms of what they're thinking when they're answering a question. I know I didn't go through each question like individually, like actually writing what I'd put in the exam, but I just thought in a subject like biology, that video would be so long and exams are so close, so I kind of wanted to shorten it down. I feel like it would have been easier to do for like a maths or a chemistry, which again, chemistry I can do if you want me to, just put it in the comments below. But I hope that was useful. It's my first time doing a video like this, so okay, if there are any like tips that you kind of think I could maybe do this and it would be better, please put them in the comments below and I'll try and adjust if I do another video like this. So, hope it was helpful. Best of luck with your exams. If you found it useful, please like the video, subscribe to the channel. We're doing really well at the moment. Any questions about what I've said or like any clarifications that you want, put them in the comments below. I'll do my best to answer. We've got our TikTok and our Instagram, both at demola. Links should be in the description below. But that's all from me. So good luck with the exams, and I'll catch you in the next one. Bye for now.